Hello, everyone, and welcome to today's webinar. My name is Catherine Heldy, and I'm the Chief Scientific Officer at SYNGAP Research Fund. Presentation today is by Dr. Michael Boland, and I have the pleasure to introduce him. Michael Boland, PhD, is a cellular and molecular biologist with expertise in human stem cell technology, transcriptomics, epigenetics, developmental neurobiology, gene-targeted therapy development, and drug screens. As an assistant professor at Columbia University Irving Medical Center, his group applied many cutting-edge approaches to study several developmental and epileptic encephalopathies, malformations of cortical development, and congenital disorders of glycosylation. His focus shifted to developing gene-targeted therapies for STXBP1 haploinsufficiency when his oldest son was diagnosed with STXBP1 disorder weeks after birth. Recently, Dr. Boland was recruited to be the Strategic Director for Translational and Clinical Research at the Center for Epilepsy and Neurodevelopmental Disorders, or END, at the Perelman School of Medicine at the University of Pennsylvania and the Children's Hospital of Philadelphia, CHOP. The mission of END is to bring novel targeted therapies for STXBP1 and SYNGAP1 disorders to the clinic, and that will be the subject of his talk today. He interacts with and advises several preclinical research groups to advance gene-targeted therapeutic strategies for SYNGAP1 and STXBP1 disorders. He also works closely with patient advocacy groups and with clinical research teams at CHOP to better understand the phenotypic heterogeneity among the STXBP1 and SYNGAP1 patient populations, and to develop molecular and physiological biomarkers as readouts of treatment efficacy in future interventional trials. Boland obtained his PhD in biochemistry and molecular biology from the University of Nebraska Medical Center in Omaha, Nebraska, and he performed his postdoctoral studies at the Scripps Research Institute in La Jolla, California. A recorded version of this webinar will be available on the SRF website at Webinars under the News and Events menu. At the end of this presentation, Dr. Boland will answer questions. We'd love to hear from you. Please write questions in the Q&A. For those of you just joining us, welcome. And again, our speaker today is Dr. Michael Boland and his presentation on END and CHOP at University of Pennsylvania. It's now my pleasure to turn things over to Mike Boland, PhD.
Thank you, Catherine. Thank you for that very thorough introduction and for the invitation to talk to all of you about the research that's happening at END. We have a lot of really exciting preclinical and clinical research going on. The focus of my talk will largely be on preclinical research because a colleague of mine, Jill McKee, will speak at a later webinar about in-depth clinical research at END related to SYNGAP-related disorders. As Catherine mentioned, I was previously at the Columbia University Medical Center, where I was studying neurodevelopmental disorders with an emphasis on pediatric epilepsies. We studied a number of different monogenic disorders, including GRIN2A, HNRMPU, GNB1, KCNT1, congenital disorders of glycosylation, such as SLC35A2, NGLY1, DPAG2, and PMM2. So we used a lot of single-cell transcriptomics as well as patch clamp electrophysiology and multi-electrode arrays to characterize developmental and functional phenotypes in many of these different disorders. It was during this time that my son Lucas was born, and we noticed him having seizures the day after birth and quickly got a genetic diagnosis that it was a frame shift in STXBP1, a gene that I hadn't studied, but actually had written a grant on the year before Lucas was born. And actually, had that grant been funded, I would have been studying STXBP1 when he was born. Lucas is 6 years old now. He remains nonverbal and non-ambulatory, but he's making great strides, walking with a walker, and I actually think he'll be ambulatory by this time next year. He'll more than likely remain nonverbal, but he's getting pretty adept at using an AAC communication device, which allows his personality to kind of come to the forefront. And let me tell you, he's a pretty sassy little kid. He's got some things to say. So it's going to be interesting to see how he develops. Today, I want to give you an overview of the END Center, and then I'll talk a little bit about some of the disease models we've developed to study SYNGAP-related disorders. I'll give you an idea of all of the different gene-targeted therapeutic modalities that we have in play at END, and discuss a little bit of molecular biomarkers at the end of the presentation. So END, or the Center for Epilepsy and Neurodevelopmental Disorders, was established in early 2023 by a large philanthropic gift to Penn Medicine under the direction of Ben Prosser. The goal, as Catherine alluded to, is to develop and bring novel therapies for the treatment of STXBP1 and SYNGAP disorders to the clinic within 5 years, and we feel that clock. We've already been around for about a year and a half, and we've made tremendous progress in a year and a half, and we still have our foot, our foot on the gas pedal. So there's absolutely no letting up. Things are actually speeding up. We've put together a really terrific team of people to achieve this goal. As I mentioned, Ben Prosser is the overall director of the center. Ingo Helbig is our director for clinical research. He has an incredible and large team of clinicians, genetic counselors, and data scientists that oversee or carry out a lot of the clinical research, which frankly is very difficult to do. We have Bev Davidson is the director of our translational research, and if anyone knows Bev, they know she's an absolute force and powerhouse in the field of gene therapy. Liz Heller, who is the aunt of a Syngapian, is one of our faculty members. I'll talk a little bit about her work. Deb French is the director of the CHOP Stem Cell Core Facility, and that group oversees all of our iPS cell generation, differentiation, and characterization. And so together, this is a really great kind of cross-functional interdisciplinary team that we really do work as a team. It's a really terrific atmosphere to be in. So if you think about bringing a therapy to the clinic, you have to think about more than just the therapy, because you have to identify and nail down what the clinical heterogeneity is in your patient population. Who are you going to treat with said therapy? What will said therapy look like? What proportion of the population will it treat? And so there's a number of different aspects that go into trying to get something into the clinic. And there are two main arms: translational research or preclinical research, as well as the clinical research. Translational research can largely be broken down into disease model development, disease-specific relevant model development, and then pre-IND and IND enabling studies. I'll talk mainly about this today. Like I said, I will leave clinical research and our large natural history study for Jill to speak at a different later webinar. But I'll talk a little bit about some of the biomarker research that we're trying to do to help in any future interventional trials. So no disease model is perfect. They all have pros and cons. So it's important to develop a number of different disease models. And when you are developing disease models, you have to realize that there's a trade-off between biological complexity and granularity, or the ability to really hone down on molecular mechanism. So obviously, genetic mouse models are extremely useful. We can test behavior. We can test how a given therapeutic agent acts in a living animal, do toxicology studies, what have you. But many of the therapies that we're interested in developing have a human-specific component. So it's important to develop human-specific disease models, and this comes in the form of human-induced pluripotent stem cells, which then can be differentiated into different neuronal cell types in the brain. Most people generally use cortical neurons because they're the easiest to generate, but you can generate cortical organoids, neuronal networks, and study even single neurons. Each of these disease models has a number of different techniques and modalities that we can use to study and identify phenotypes within that disease model. I won't go through all of this today. I'm just going to highlight a few examples of some of the work that we're doing with the disease models we've developed. So at end, we realized that in order to test human-specific therapies in an animal model, we wanted to develop an animal model that has a humanized SYNGAP gene. And so what we did was we humanized the entire locus of the SYNGAP gene in a mouse. This includes all of the upstream and downstream regulatory elements for SYNGAP. So Ignalia and Brandon Brown are currently characterizing EEG and behavioral phenotypes in the humanized haploinsufficient mouse models for SYNGAP. We did do some molecular characterization of these mouse models to demonstrate protein levels of SYNGAP and mRNA levels. And so at the top here, you can see we have mouse and human specific TACMAN probes that detect the different transcripts respectively. So in the ++, which is a wild type mouse, you see 100% SYNGAP levels. In the hybrid animal, which has one humanized allele and one mouse allele, you have 50% of the mouse transcript, and in the human, you have none of the mouse transcript. And that's the opposite for the humanized specific probes, where the humanized animals express all human mRNA and none of the mouse allele. Conversely, on the bottom, what you can see here is we've characterized the haploinsufficient humanized mouse, and we can demonstrate that, yes, it is truly haploinsufficient. It results in a 50% reduction of protein. Now, I will say that this humanized haploinsufficient mouse model is breeding the humanized mouse to a mouse haploinsufficient. So the minus allele is a mouse allele. That's the knockout. We also have developed some patient-derived iPS cell lines and generated isogenic controls using CRISPR-Cas9 genomic engineering. We have a K1185X mutation from a little boy and an R1240X from a little girl. If we look at the variant spectrum across SYNGAP, one thing that's really striking is the preponderance of truncating variants versus missense variants. If you look just at the variants presented here in this review by Rick Hugenier's group, you got about 80% protein truncating variants that include frame shifts, deletions, nonsense mutations, what have you, versus 18% of missense mutations. We wanted to develop an allelic series for SYNGAP so that we could test any therapy across multiple genetic variants on a uniform background. So what we did was we took an unaffected iPS cell line, developed at CHOP, and is really extensively quality controlled by CNV, karyotyping, as well as differentiation. And this line is absolutely terrific. It has almost no CNVs, differentiates very well into multiple different neuronal subtypes. And so what we did was we used CRISPR-Cas9 to knock in a number of variants across the SYNGAP gene including 2 missense variants that are recurrent across the patient population. So now that we have this allelic series, we can begin to characterize some of the functional phenotypes that occur with each of these variants on a uniform genetic background and then test therapeutic modalities on this. So I'll switch gears a little bit to talk about the preclinical research. I'll give you examples of 5 different gene-targeted therapies that we have in development. This is gene replacement, antisense oligos, CRISPR activation, tRNA read-through, and protein stability through engineered deubiquinases. And you can see all the different groups here at end that are involved in this type of research. So Bev Davidson's group has been working for years to develop really terrific AAV capsids that have been evolved in non-human primates. They have capsids that can target even specific brain regions and capsids that show really high transduction efficiency across the non-human primate brain. For SYNGAP, we want something that's widely distributed across the brain and definitely want something that targets excitatory neurons where SYNGAP lives. So we're working on gene replacement and then packaging this in Bev's fancy capsids to test in animal models like our humanized mouse model, which these capsids also show really high transduction efficiency and broad biodistribution in mouse models and rat models as well. So this is a really terrific tool for all of the AAV-mediated gene therapies that we're working on. In Ben Prosser's lab, we're starting to look at the different isoforms of SYNGAP. And as most of you know, SYNGAP has a complex alternative splicing structure, and it results in a number of different N-terminal and C-terminal isoforms. What we're interested in is the biological effects of each of these isoforms during development and their ability to correct functional deficits in neurons that lack SYNGAP. And so we're utilizing our humanized SYNGAP mouse model, cloning different isoforms into lentiviruses, transducing primary neuronal networks that were derived from the SYNGAP humanized mouse models, and then we'll be testing cellular morphology such as dendritic arborization. We'll be looking at downstream molecular events. We'll be looking at functional correction via multi-electrode array and trying to determine which of the SYNGAP isoforms may be the most suitable for therapeutic correction. I'm sure many of you know that there are many ways to treat haploinsufficiency with an ASO. You can target the 5' regions and try to enhance translation, initiation, or elongation. You can target the open reading frame of the gene to induce, say, splice switching to create more productive splicing of a transcript. You can also target the 3' UTR of the gene in a number of different ways, one of which is steric blocking oligos that would presumably inhibit microRNA or ribonucleotide protein particles that are negative regulators of either transcription or translation. And Ben Prosser's group has been working for a number of years on many of these different strategies. And just last year, they published a really nice study where they identified PTBP2 binding sites in an intron of SYNGAP1, and by inhibiting this, they could actually by inhibiting PTB binding with an ASO, they could initiate splice switching and more productive transcript production of the SYNGAP gene. So this is one example of an ASO that can restore STXBP1 or STXBP1, can definitely edit that out. This is one method to restore SYNGAP levels in models of haploinsufficiency. The other way that Ben's group is looking at this, and one of their favorite methods, is through steric blocking. And in collaboration with Ionis, they've done many walks across different regions of the SYNGAP 3' UTR, and they've identified a few regions where known microRNAs are bound, and they've identified some really promising lead candidates that are able to upregulate both SYNGAP mRNA and some that upregulate protein in induced pluripotent stem cell derived neurons. And so this is a really active area of research at End. Another really active research at End, and one that I really like, is CRISPR activation. And Liz Heller's group is pioneering this for SYNGAP, and they've developed a CRISPR activation strategy based on SP-DCAS9 that is fused to the VPR transcriptional activator. And they've got a number of different models that they can work with to test these different constructs. So they've developed CRISPR activation constructs that work in the mouse and CRISPR-A constructs that work in humans, because the promoter regulatory regions of the two species are different. So you have to design different CRISPR-A constructs depending on what species you're in. And they've actually done both and have shown efficacy in both. So I'll show you a little bit of a piece of data from Laura Sicklinger and Molly Riley in Liz's group, where they've designed some guide RNAs to the mouse region of SYNGAP, and they used epigenetic modifications such as trimethylation of H3 lysine 4, which is a marker of open chromatin. And so they looked at SYNGAP and were able to identify regions of open chromatin based on these epigenetic modifications and designed guide RNAs in this region. Then they packaged this into a lentivirus and injected one half of the hippocampus with SYNGAP guide RNA, and the other side of the brain, the other half of the hippocampus, with a control guide RNA. And this is done in a mouse model of SYNGAP haploinsufficiency. And they're able to upregulate SYNGAP protein to levels of wild-type mice. So this is really, really exciting, and we're testing these constructs in mouse models for phenotypic correction of behavior and functional rescue at the same time. Another modality that is really exciting and has the potential to not only have a huge impact on SYNGAP, but also on almost any other neurodevelopmental disorder that results in a protein truncation variant from, say, an arginine in this case. This is essentially an engineered, modified transfer RNA that, when incorporated into the ribosome, can actually read through the premature stop codon and produce a full-length transcript. And so we've developed a couple of different of these ACE tRNA constructs, and Alex in Ben's lab has been shown or has shown a dose-dependent increase in SYNGAP protein using this method. And we're continuing this work in other cell lines and are eager to generate the R1240X mouse on the humanized background, which is actually underway at at an industry partner, and we'll be able to essentially test this therapy in an in vivo model. The other gene-targeted therapy that we're looking at is through increasing protein stability. There's a few pieces of evidence in the literature that SYNGAP is ubiquitinated, which results in protein degradation. So we're trying to use engineered deubiquitinases to specifically inhibit ubiquitination of SYNGAP and thereby increase its half-life. So this work is in collaboration with Flux Therapeutics, also in Ben Prosser's group. So in the last few slides, I want to talk a little bit about clinical research because it's a huge part of END. But like I said, I will leave the details to Jill in a later webinar. But a big part of END is development of studies to define the phenotypic heterogeneity in patient populations. We're doing this through retrospective and prospective natural history studies. The prospective natural history study that we have going on right now, I'm sure most of you are aware of, is this multi-site natural history study. CHOP is the data coordinating center for this, and we've got 2 sites up and running and ready to see SYNGAP patients, Colorado Children's Hospital as well as Stanford Medicine. And it's my understanding we'll be adding maybe 1 or 2 more sites in the future. But so far at CHOP, just in the last year, we've seen 87 SYNGAPians. And this really, I just looked at the breakdown of gender versus variant type and age group, and it really does have a really good broad distribution across the SYNGAPian population in terms of age and variant type. But I want to take a few minutes to talk about biomarkers because this is kind of the holy grail, something that we've talked about doing for years. And now that we have the means to do it, we're doing it. And it comes in 2 flavors: physiological biomarker development. We're looking to develop disorder-specific EEG fingerprints. And through work that Jill and Peter have done, we are already starting to identify STXBP1 and SYNGAP specific EEG fingerprints. You can see a famous little SYNGAPian here with a EEG cap on. He looks great with that cap on. The other thing that we're looking at is sleep and sleep disturbance as a really solid potential outcome measure for future interventional trials. And we've recruited a renowned sleep researcher, Maya Bushan, to absolutely nail down sleep patterns in SYNGAPians. And the way that we're doing this is we're essentially, Maya has a really large sleep study, and we're enrolling our patients from our natural history study into her sleep study through a consent. And what she does is she just sends families this watch, which has actimetry and accelerometry and a number of different measures that can be used to accurately record sleep. And she sends the family the watch. The child wears it for 3 weeks, and you send it back, and they get the data. It's that easy. So this is up and running. It will be up and running for our natural history study very soon. The other aspect of molecular biomarker development And this is something that we're really interested in as well, because for any interventional trial, you want or hope to have a biomarker that can predict uh, target engagement and may also give you an idea of therapeutic efficacy before you get to downstream outcome measures like behavioral modification or what have you. And so we're really digging into proteomics of CSF and some blood and plasma proteomics. So we've teamed up with the Center for Diagnostic Innovation, led by Steve Master. We're doing some targeted mass spec to look at tryptic peptides for SYNGAP, either just without enrichment or following antibody enrichment or looking at extracellular vesicles. With Lynn Spruce's group at the CHOP Pen Proteomics Core, we're taking an unbiased mass spec approach. This is essentially a proprietary milieu of 5 different nanoparticles that have different protein binding affinities. And so we're able to take an unbiased approach and look at um, essentially the proteome within the CSF sample rather than kind of a more targeted approach like what we're doing with Steve Master. Recently, SRF has contracted Olink to look at SYNGAP levels and develop a proteomic profile of a number of different, I think it was 76 samples of Syngapians and familial controls. And Janine in Ben Prosser's group did the data analysis of this Olink data, and we were able to show that SYNGAP levels are below the level of detection in these samples. This is the level of detection here. There was really no difference between familial controls and proband, and the levels are so low that it's not reliable as a marker. But interestingly, we were able to find one protein that is significantly upregulated in the patients versus their familial controls. And you can see kind of a better distribution of this on these lollipops down here, where you've got SYNGAP, where you really don't see any specific upregulation in proband versus control. It's kind of all over the place. Whereas with this protein X, there is a very significant upregulation of this protein in patients. And if you look at a volcano plot of essentially all of the proteins in these samples, somewhere around 5,400 proteins, this protein X really stands out as upregulated in Syngapians. And so we're very interested to follow up on this. In CSF samples, and also to see if we can change the levels of this protein after SYNGAP rescue. This protein is really, really interesting because it's a hormone involved in hyperactivity, anxiety phenotypes in animal models. So this is what clinical trial readiness essentially looks like after you put all of the pieces together. This is what we're trying to do at End, and we're making significant progress in every one of these areas. And so I'm really confident that we're gonna have that timeline of 5 years it looks like it's real. With that, I'll end with a kind of outdated photo of End. There's about double this many people now at End. This was at the launch of our center in May of last year. And I will open it up to questions.
Thank you so much, Mike. That is that's a lot of work, and I knew about some of it. I didn't know about some of it. And so this is why it's so important for us to have you here and explaining what's really going on. Um, let's see. I don't think there are any, yeah, there are no questions in the Q&A yet. We'll see if anybody wants to join in there. And let me just change my view. Um,
I
yeah, so
can I
yeah, so
ask just
Mike, yeah.
an easy question just to get the ball rolling? That protein X in the O-link data, and I'm thrilled to see that we already have such a meaty finding so quickly because we just shared that data really recently, right? So that's
Right.
exciting. That protein X, you said we're going to look for it in CSF. So because is it worth looking for it also in patient blood, or we just think it would be too hard to detect?
This is from patient blood.
Right, but with a really expensive machine to do the assay, right? So I'm looking for something that's like we could use in a trial as a biomarker.
No, 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 absolutely, Mike. That's exactly what we're thinking.
Cool.
So we're not only interested in looking at like SYNGAP for target engagement, but we also want to be able to look at some of these other proteins that are coming out in the analysis. So this protein X, um, even if it's not in CSF or
Mm,
detectable in brain tissue, um, we can still use this as a potential biomarker in patients. So one of the things that we're really interested in digging into is this protein was also detected in mouse
right.
blood plasma. So now we have a model that we can start to test hypotheses on. You know, we can look at CSF and brain tissue from the mice, see if we can see upregulation of this protein in that tissue. And even if we don't, that's okay. What we can then do is see if we can correct SYNGAP levels in the CNS, does that have an effect on this protein in blood plasma, which would ultimately be like the holy grail. That would be almost a biomarker of efficacy and target engagement.
But it's also, yes, cool. And you sort of you sort of led me to this next question by noting that this protein has been associated with hyperactivity and anxiety in other mouse models. I mean, that's hyperactivity and anxiety are things that plague us and our kids, right?
Yeah,
It might plague some of us, but as a result of plaguing our kids, it plagues the parents. So
yeah.
does that point us to like some kind of mechanism of action for disease and maybe suggest other therapeutics in the meantime, that not to just go to the root cause, but to help treat our kids' hyperactivity and anxiety as a second order thought?
It's possible, absolutely. So one of the things that with any therapeutic modality, you want to either go to the root cause or frankly what we've been doing for years is treat symptoms
Right.
or try to treat symptoms.
Well, that's what we're
Um,
stuck with until one of those things you had up works, right?
yeah, that's exactly right. But I'm really hopeful that we're going to have something at the root cause
Right.
sooner rather than later.
It's amazing.
Then, going with that question, my question is the ambition to get maybe CSF samples from patients that is a little bit more difficult. I like your point that you guys are gonna try to see it in plasma because CSF samples imply a lot of moving parts.
Yeah.
The risk of a lumbar puncture on
Yep.
patients, then it's gonna be hard. Then I like the plasma idea better than the CSF, but do you have any samples of CSF of
Yes.
patients, Syngap patients there?
Not SYNGAP patients. We would have to include that. So we've already amended our natural history study protocol to do elective spinal taps.
Mhm.
Um, we have a bank of CSF at CHOP and at Penn Medicine from normal. So what we can do is we can look to see what I really want to do is for any of the CSF proteomics, I want to exhaust normal samples before we ask patients and kids to undergo spinal taps. So I want to have something that we can reliably detect in normal CSF before we would go into a patient. The name of the protein, I'm not sure I can divulge that in the webinar. I can divulge it to you, Marta, afterwards. But
Thank you.
it was independently confirmed by a group at Baylor as well.
You know,
So
it's exciting. That's
it
really cool.
is. It was striking that it's so much different in probands.
And just for the non-scientists watching, I mean, I love that you kept this heavy, but the proband is the patient, right? And then
Proband is the patient. Yeah.
right. And the other thing you didn't say that as one of the non-scientists on this call, it was helpful when someone explained it to me, because I remember way back when we began conversations about this humanized mouse, because these things were not cheap to make, and we're thrilled that you guys paid for that mouse. And now you're paying to characterize it, which is the more expensive part. The reason we needed a humanized mouse, because that's a new thing, right? That's like the new cool thing. Every rare disease group wants a humanized mouse. Nobody had these until recently.
Yeah.
And I was like, why do we suddenly need a humanized mouse? And please correct me, but the answer is because because the humanized SYNGAP and the mouse SYNGAP are same, but different. Like there's minor differences.
Right.
And some of these therapeutic modalities focus on things that exist in the humanized gene, but not the mouse gene. So we needed to stick the humanized gene in the mouse so we could test those modalities in a living creature that wasn't a human.
That's exactly right. That's
All right.
exactly right. That's exactly right. So the protein overlap between mouse and human is very high, but the regulatory elements that regulate how SYNGAP is expressed are different. And many of the therapeutic modalities, like these steric blocking oligos or CRISPR-A, target regions of the human gene that are different from the mouse. So we needed a human gene in the mouse to test this inside of an organism.
Got it.
Yep, that's exactly right. And I will say that Brandon has done some of the behavioral characters, started to do a lot of behavioral characterization of this humanized mouse and the haploinsufficient mouse. And it very closely, it replicates a lot of what's been seen in the literature for other SYNGAP models of haploinsufficiency, hyperactivity, things like that. So I think we have a legit humanized mouse model for SYNGAP. For STXBP1, no, that model is not very useful. But for SYNGAP, it looks like it's legit.
I want to encourage because there's a lot of smart people on this call who are probably have great questions, and I want to encourage them to throw them in or raise their hand, and we can make you panelists. But I think one of the things that's fascinating to me about END is that you're looking at two synaptic, one's presynaptic, one's postsynaptic genes in parallel. Like everything's sort of been trying to be done to both. And I'm just curious, like, have there been any learnings? Has that facilitated any learnings by looking at the same experiment in two different genes that are same, but different, and trying to like tease apart? Has there been any synergies for that? I guess that's my question.
There really haven't been synergies for that. It's a really good question. The phenotypes are different. So even when we do something like MEA or something like that, the phenotypes are different. So what we want to do is we want to essentially develop these different disease models and look at disease-specific, disease-relevant phenotypes. For instance, as you know, SYNGAP and the redistribution of AMPA receptors or downstream signaling through ERK. We want to, or some of the morphological defects that have been observed in dendritic arborization of neurons. Those aren't seen in STXBP1. Those are specific to SYNGAP. So we're trying a lot of the same modalities because they're both largely haploinsufficient disorders. Granted, there undoubtedly will be some missense variants that have other modes of action, but for the most part, both of these are haploinsufficiency. And so we're trying to restore protein levels for either STXBP1 or SYNGAP, but really in a kind of a disease-specific context.
Got it.
There are two questions in the Q&A from Hans. I don't know if you can see those yourself, Mike, or if you want me to read them to you.
No, I can't see him.
Okay, so Hans says his first question is,
Oh, no, I can't see it.
okay,
Sorry.
great. Why don't you go for that?
Hans says there's a dedicated blood test for protein X at the Mayo Clinic. That's absolutely terrific. Thank you, Hans. This is why we need you on this call. Uh, we just got the results of this just a couple of weeks ago and haven't really dug into it. I'm glad for that answer. The other question is, yes, there's concern that overexpression of SYNGAP is toxic to neurons. This is something we're well aware of, and it's not like this is a situation that is more like MECP2 than STXBP1, where it seems like STXBP1 has a natural break on protein expression above wild type. We're very well aware of this overexpression. Luckily, what we see with the CRISPR-A is we've never seen overexpression of SYNGAP. Now, it will be interesting to see what we get when we start really digging into the gene replacement strategies. But yeah, we're definitely aware of this. The phenotype that can result from overexpression of SYNGAP.
Yeah, in addition, oh, go ahead, Mike.
No, sorry, Catherine, go ahead.
So in addition to the overexpression, I'm wondering about the, increasing the length of time that SYNGAP is around. Like that's another way to increase
Sure.
the amount of protein, right? So I'm wondering how, I'm just wondering if you've seen, if you've seen yet some amount of SYNGAP, do you ever see it double or 5 times the amount of protein that's around? And no, okay. And then do you ever see it, there's a recent, just this week, paper on interactions between different another sort of RAS gap protein, right? So these things antagonizing each other as being important. And then there's another one talking about translation at the synapse rather than cytoplasmic SYNGAP.
Oh yeah,
So,
no, absolutely.
So I think a couple things that I didn't hear you guys talking about were subcellular localization. So that would be translation at the synapse.
Right.
Another one would be like sort of making a mini gene. Like how are we going to package this in an AAV? I heard you talk about isoforms, which is fantastic. I think in addition to isoforms, it's sort of like how do we package this in Beth Davidson's AAV? So
Well,
I assume you guys I mean, I know you guys know all these things, and so just wondering how you see them working together and what I guess sort of more what's a timeline like this if we're a year and a half into 5 years. Are you thinking about clinical trials at year 5? Are you thinking about clinical trials before year 5?
I mean, I hopefully it's before year 5, but I can't. I really don't want to make a strong prediction and be
Right.
wrong. All I can say is that we have a lot of balls in the air,
Yeah.
and one of these is gonna show fruition. As far as packaging in an AAV, full length A alpha 1 SYNGAP fits in an AAV. It's 4.1 kb. The packaging capacity of a AAV is about 4.8 kb. So you can get full length SYNGAP with a promoter into an AAV. Now, the packaging capacity that might get you towards the upper limit of the packaging capacity And so we really would like to know if some of these other isoforms can functionally compensate for, say, A alpha 1, which is the longest isoform. We just won't know that. We're starting those experiments like right now.
And what do you think about potentially needing multiple isoforms to interact with each other? I
Yeah,
mean,
I
is the
mean
Holy Grail really getting one, or what if we need two to sort of?
I nobody really knows. There's some evidence that these isoforms are developmentally regulated.
Okay, so by the time you're treating, there's a good one for that moment.
Yeah, so that's why we have the two different models. So we can start to look at isoform expression during differentiation of iPS cell neurons. We can look at isoform expression at different stages during mouse brain development, things like that. Um, so it's a really good question. The isoform question is a big one, and we're that might take a little more time than something like ASO or CRISPR-A.
Right. Okay.
Yeah. And like Hans astutely pointed out, we have to worry about overexpression as well.
On that, a clinician called me last week and said they had a patient who had a full duplication.
Yep. We
And
have one.
yeah, and
Yeah.
they're human, alive, and well, and as you might expect, unlike our kids who are hyperactive and can't sit down, this one's the opposite.
Yeah.
Right.
It's a distinct, it
Yeah.
looks like a distinct disorder. Yeah.
So
We have one at CHOP too. We got blood to make iPS cells from that patient.
that's great. So that would be huge because this question of overexpression, I also get SRF gets a proposal once a year from someone who wants to build a model somewhere between 6 and 7 figures to test this issue. And then industry is like, just wait for us to do it at a safety level. Like don't get in front of us and create more problems. So it's a really interesting bugbear that it's one of those questions people love to ask, but I'm really glad now to hear from two different sides of the country that we're finding humans who have extra SYNGAP and
Yep.
turns out they're alive.
Yeah, they essentially have 3 copies of SYNGAP.
Right. Pretty exciting.
Yeah, it's really cool. My jaw hit the floor when they told me at End Clinic, it was about 5 or 6 months ago, about this patient with a duplication. I said, get blood.
Yeah.
Yeah, go ahead.
So that's actually a good question. We have a biorepository that's some people are in, and I'm wondering, does CHOP have a SYNGAP biorepository associated with the NHS?
Yes, but we haven't been systematically collecting for in either of the natural history studies.
So as 2025 comes around and your CSF protocol kicks in, would you potentially be doing CSF and blood at that point?
Yes.
Yeah.
Yeah. And the fact that we see this protein X in blood plasma from Syngapians makes it even more attractive to start collecting blood. Yeah, definitely.
Right. Okay, fantastic.
And we should all look into that Mayo test too and see if we can.
Yeah, definitely. I'm glad Hans pointed that out. I will look into that.
That's amazing.
Do you have any questions for us, Mike? Mike Boland, do you have any questions that anything that you're hoping for?
No, just to keep up the amazing work that you all are doing. We saw 87 patients at one clinic in a little over a year. That's insane. And that's because of the SYNGAP community and the ability of SRF to recruit and put word out. I mean, we didn't see that many STXers at CHOP. We saw far more Syngapians than we saw STXers at CHOP. So hopefully Charlene's not watching, but the SYNGAP recruitment was way better at CHOP.
First of all, Charlene hears everything. Second of all, you gotta be careful with that.
Yeah.
And in fairness to STX, you guys had already been up and running and have multiple other clinics, right? So we
Yeah,
are
but
we're
but
playing
the
catch up.
only clinic that really saw substantial patients was Colorado. Baylor and Weill Cornell took a little bit longer. They've seen a handful of patients at both, but it was mainly CHOP and Colorado.
Now you've mentioned Weill Cornell. I might have blinked, but did you mention EEG biomarkers? Because is that also something END is looking at?
Yeah, absolutely. So I showed a
That was the
photo
picture.
of
That was the picture of Tony, right?
yeah, yeah.
But
So
so I have a question on that. I'm sorry. Because what we're doing, I keep explaining this to parents. So like, well, but EEG, blah, blah, blah. I'm like, the QEEG that they do in clinic is way different. It's like a high-powered telescope as opposed to like a dinky little home telescope. Way different than the clinical EEGs.
right.
When you find, I think I like how you coined it, the SYNGAP fingerprint in that QEEG, is the next step to go and see if that same fingerprint is detectable in the regular clinical EEG?
Yeah, we can look at that. So that's where Peter's work, he started with clinical EEGs,
Got it.
and now we're digging into the quantitative EEG. That's what Jill is going to be working on. So the nice thing about the QEEG is you've been there. It's only a 15-20 minute recording, and we actually don't even need all of that recording. We just
Right.
need snippets of it because it's 128 electrodes versus like 35. So I didn't want to steal Jill's thunder. She'll be talking more about that at a later webinar and certainly at the SRF meeting right before AES.
Amazing.
Yeah, I think EEG is something that every industry sponsor is interested in. It's the lowest hanging fruit because it's the most quantitative. Um, Easier to quantitate than behavior or something like that. And that's why I'm actually really interested in sleep because I know you guys the community has such an issue with sleep that that's another thing that's quantifiable. And if there's a therapeutic agent that does show efficacy, that is something that you should probably be able to pick up on relatively quickly, like an EEG phenotype, before you can maybe modulate behavior or quantifiably modulate behavior. Marta?
I was still running my head through the duplication patient, and that we need to find a mechanism to group the 2 or 3 patients of duplication and try to phenotype those patients.
Yeah.
Uh,
It's really interesting. Some of these other monogenic neurodevelopmental disorders um, you can almost parse them out by mechanism of action, like SETBP1 is one of these, where there's a gain of function, there's a loss of function. You would almost consider this duplication a gain of function for SYNGAP because you have extra levels of it that create a completely different phenotype from what you see in loss of function haploinsufficiency. You're absolutely right. It's fascinating to know that there's at least a handful of these patients out there, and there's got to be more.
And then the question is, how are we not finding them? And I wonder if the answer is because the gain of functions are just so much milder and that's just a hypothesis. I haven't met either of them, but
Yeah, it could be that it's more mild or
many other possible answers.
yeah, I mean there's so many possible answers. I know Jill has a patient with the duplication.
That's great.
Maybe 2.
I have one last question, unless Yulia or Craig in the chat, if you guys want to chime in, but like, if you follow my podcast, I'm obsessed with how many papers come out on SYNGAP papers aren't going to cure kids, but it sure is a great proxy for scientific activity. This year we're going to set a record for the most papers. Last time I checked, I think we're at 43. If you had to guess based on everything you just put up, how many papers is Chop Pen alone going to be publishing next year? Just like each of those slides sounded like a project that could result in the public. I have another question after this too, but
Absolutely. I think in the next year or two, there's going to be a flurry of papers. I mean, there's already one from Prosser's group.
yeah.
Jill's work on the retrospective natural history study, mining the citizen data and all of that is under review right now.
Yeah.
So yeah, we kind of just started doing this about a year and a half, 2 years ago, and I think that yeah, in 2025 or early 2026, there's going to be a flurry of papers. And frankly, I'm with you, Mike. Papers don't cure kids. They don't treat kids. So I'm at the point now where I'm like, yeah, papers are great, but it's not the be all and end all for what we really want to do. Yes, it helps us. Papers will help us attract additional funding and whatnot once because the end funding isn't going to last forever. But we we have a clear mandate and means to achieve that, I think. And that's really high on all of our minds.
That's great.
Um,
Go ahead,
A
Catherine.
follow-up to that with the papers is, does END have a web page that sort of shows deliverables? Do you have a
Yeah,
page
we have a website that has publications and things like that on it.
that have come from this donation, this philanthropic donation?
Yes, yeah.
Awesome. Okay, we should
Like we have the bioarchive paper for the
yeah,
mouse models. We've got Ben Prosser's PTBP2 paper.
great,
We'll have we've got some STXBP1 papers. We'll have SYNGAP clinical papers in the next 6 months. So yeah, there all of that. Anytime we have something that's related to end, we put it on the website.
great. Okay, thank you.
Here's my second question. There's a I won't bring that up, but sometimes parents see papers about mice being cured from SYNGAP and they get really excited and they find this paper and they send it to me as if I hadn't seen it yet, and then they say, well, when are we going into human with this?
Yeah.
And I mean, there was that the one I was not going to bring up was there was this one out of Davis that had a very problematic promoter, and I was like, this is not going to be anywhere near human for a long time. But like, there are some things that prove concept but don't translate. And if I understand correctly, the CRISPR-A work out of Heller's lab that you shared, that's incredibly exciting. But because of the lentivirus it's using, it's not really translatable right now. So what in that case, like, what is the next? How would that go into humans if we were to take that proof?
Well, we would just, I mean, the guide RNA, we could switch that into an AAV.
Okay.
So they're using lentivirus because it's easier to screen guide RNAs. But once they show efficacy, we can use either a split AAV where one virus has the dead Cas9 and the other one has the guide RNA scaffold. And when I was at Columbia, I developed a single AAV. It's based on a different species of dead Cas9. A different bacteria that it was a different dead Cas9 that's smaller, but you can fit everything into one AAV.
Awesome. Okay.
So yeah, it's you're absolutely right. You can't use the lenti, but we can easily adapt that to an AAV.
It's incredible. Are you gonna have a bake-off at some point? Like, if you have a few promising things, we're just gonna treat like 5 different mice and see which one does best?
Yeah. That's I mean, that's kind of the idea because none of these therapies are one size fits all. And, that question brings to mind Alison Berendt when she shows all of the different academic labs that have different mouse models and they're testing gene replacement or ASOs or CRISPR-A or whatnot, and which level of correction of different molecular rescue and functional phenotypes and behavior and all of this stuff, each of these different modalities has a different flavor in an animal model. And so, and it's probably going to be the same way in kids, unfortunately. So, it's a big black box. I mean, and this is something that the entire field is grappling with. What would be the most efficacious therapy? And at end, we take the view that if you can restore the protein levels back to wild type levels, that should take care of the phenotypes. And if you look at that Allison Barrett, I'm not going to do a plug for CRISPR-A, but if you look at that slide, CRISPR-A corrected almost everything. It was green. She had green for if it corrected a phenotype, or red if it didn't. And ASO had 50-50 gene replacement, 50-50 CRISPR-A, green the whole way down. So time will tell, and frankly, Mike, it could be disease specific too. You know, it's not one of these therapies isn't the type of therapy may matter for the disorder. Like Hans said, gene replacement may not be the right way to go at it because overexpression is such an issue. So it could be that you need something like CRISPR-A or ASO that can bump the levels back up to wild type, but you don't have to worry about going too high.
Cool.
Yeah.
Thank you. I'll give it back to you, Catherine, but thanks, Mike. This was really helpful to have it all in one place.
Yeah.
I'm glad to do it. We've got a lot of stuff going on.
Yeah. I just have one last sort of science thing before we wrap up. And that is, in addition to these therapeutic modalities, there's always, where do they go? Which cell types? And so I noticed you talking about the excitatory cortical neurons, but it seems like there are quite a few cell types that have SYNGAP. And I'm just wondering who is doing, who's got their finger on the pulse of like which neurons at a stage where we'll be curing kids, like at that not the earlier stuff, which of course I'm fascinated by as a developmental biologist, but just focusing on therapeutics, who's looking at which cell types are really required in order for things to get better.
So Ingo's group has some guys that are mining RNA-seq data, single cell RNA-seq data. We know that there are likely other cell types other than glutamatergic excitatory neurons in the cortex that express SYNGAP1. Most of the available evidence based on SYNGAP's interaction partners, like DLG4, PSD95, those are found in excitatory synapses. So we can reasonably expect that reestablishing SYNGAP in excitatory neurons will correct phenotypes in a mouse model.
And that's for the synaptic plasticity phenotype, yeah.
It should correct hyperactivity, synaptic plasticity, learning and memory deficits. It should correct all of that.
Seizures. Okay.
Yeah, it should.
I have a sense that
I think
potentially anxiety might be more. It seems just from understanding anxiety that other cells might be involved too, but we'll see. I guess we'll see.
Sure.
I've never been sure if anxiety for our kids is really anxiety or if it's sensory distress or if it's a combination.
Right. And I mean, so this is something that I talked to the Allen Brain Institute about, is designing really good, because if you're thinking about an AAV mediated therapy, it comes down to the promoter you use, where you're going to express DEDCAS9 or where you're going to express the SYNGAP isoform. And so, once I can identify, so the Allen Brain Institute has developed really good and identified really good specific regulatory elements for different types of neurons. And we could theoretically design and build a vector that could target expression of SYNGAP or DEDCAS9 or what have you to several different neuronal subtypes, even astrocytes, for instance.
Yeah.
But for the time being, what we can do is we can use something like a synapsin promoter and express it in essentially all neurons, with the thinking that if the machinery isn't there in the cell type, if the cell type doesn't use SYNGAP, having SYNGAP expressed may not have an effect.
Yeah. Yeah. Okay. Well, lots of amazing
Lots of
work.
questions. Yeah,
Well, lots of
so many
questions,
questions.
but you have so many labs and ways to look at things that it's really exciting to have all that kind of under one. I was gonna say one roof, but you might be in different buildings, but under one umbrella.
It's under one umbrella, and it's like I said, it's a really close collaborative atmosphere. If you look at the Janine's paper on the PTBB2, it was Ben, Deb French, Bev. It's like it really is a team effort. And so it's like Ben Prosser's working on ASOs, but we meet every 2 weeks and we discuss, hey, what should you try next? So it really is a great collaborative atmosphere at End. And we really are pushing to make a difference in these communities.
Well, that's great that the scientists know what each other know. That's so helpful.
Yeah, it is. And it's like, and it's really nice to have the clinical piece right there too. And so we can, learn from what they see in the clinic and vice versa. So I think that's one of the more unique aspects of End, is to have both the preclinical and clinical work together under the same roof, and we meet regularly to discuss.
Yeah. Well, thank you so much. Thanks for giving us your time. Thanks for your wonderful slides. Thanks for going over all these things and answering a lot of questions. We really appreciate it.
Thank you.
This was very good. Thank you.
Thanks.
Bringing the awareness of all this together to the community help us to explain more to the community why you need to go there, why you need to donate your blood, why all these things can come together. It's really helpful. Thank you so much.
Yeah, I mean, it literally is. We can't do this without the SYNGAP community. We can't. The patients and the families are so important to this success. It doesn't happen without them. It really doesn't. The clinic, especially the clinical research, it just doesn't happen. We could develop a therapeutic and then we're like, okay, well, who should we treat with this? What age is the most appropriate? What are the best outcomes? Without being able to, look at a wide cross section of patients, we wouldn't know. So it's incredibly important that the SYNGAP community knows how important they really are. Okay.
Well,
All right,
thank
I
you
have
so
to
much
run.
for this. Bye.
Me too.
Thanks.
Thank you
Bye.
so much. This was great.
Thank you.
I'm glad you liked it.